those, or we still have some antibodies, it's not making us sick yet. Third year comes around, and there's been one more small minor change to this molecule here, and now we're not recognizing either of these, and so this virus makes us sick. We get the flu, okay? Now we'll have immunity for this and this going forward, but at the time, we don't. Now, imagine antigenic shift would be something like this, would be something like this, where you have your virus, two of them come together. I should draw another one. Okay, so these two come together, and you're saying, well, what if you have some immunity to this one, and you also have some immunity to this one? What if you recognize this one here, and you can recognize this one here on this virus? Is that kind of what you're saying? Absolutely, you could have some residual immunity to that. It's very possible that you would have antibiotics, and that is why America probably didn't suffer as great from the most recent swine flu as Mexico did. Because of the immunity that the states had that Mexico didn't, you probably saw less virulence in the states than you saw down in Mexico because of that somewhat residual immunity. And also might be why you're seeing the older population do better than the younger population, because of that somewhat residual immunity. But back to these guys coming together. Now, if these two infect the same cell and genetic material gets swapped, you might end up with something that has a completely different combination of surface receptors. And then we just don't recognize it at all. Or a majority of the population doesn't recognize it. There'll always be a few people that are immune to any kind of disease or virus. Even the AIDS virus, which initially affected just about everybody that came in contact with, still, there are about 1% of the population that lacks the surface receptor that allows for infection to take place. So even if 90% of the population came down with AIDS, there still would be 1% of the population that would be immune to it. And that's almost the case with any kind of virus, any kind of bacterium. They'll still be able to make the majority of the community sick, but there's always going to be a few individuals that are immune, not necessarily because their immune system has seen the organism, but maybe because their surface proteins or their surface receptors can't be bound to. And so it provides them, because of a genetic anomaly, provides them with immunity. Yes?
I'm going to go back a little bit.
Okay, that's fine.
You were talking about temperate infections. You were saying that if a cell is infected, it's in a lysogenic state.
Yes.
So that's not when it lyses the cell.
It just means that that is a possibility. Right. At any time that a virus is infected, a bacterium or another cell, lysis is a possibility. The virus reproducing and causing it to lyse. And so when a virus is integrated itself into the host DNA, we say that the host has been lysogenized, So it's a lysogenic state. And that means then that virus, because of environmental cues or something, could then start causing replication and eventually cause the cell to lyse.
And is transduction only in
Yes, because the virus has to integrate into the host DNA. Most productive infections, the virus really doesn't integrate into the host. It just into the host DNA. It just infects the host, takes over the machinery, makes lots of copies until the host lyses. So yes, a tempered infection or lysogenic has to happen for a tempered infection. So it has to integrate into the host DNA.
Yeah. I just have a quick question. I was just hoping that we could just get through the review first and then we'll put this to the end. We only have like 15 minutes left, and we have a lot to go through.
Well, I told you I wasn't going to be able to get through everything. Okay, I like to focus on what needs to be cleared up, though, because it could be that I'm going over stuff that everybody understands, so I don't want to say no questions, because some things need to be clarified. But I'll try to speed it up some. Okay. Okay. So where are we? We're done with viruses. Let's move on to microbial ecology. So with this here, understand microenvironment versus macroenvironment, okay? And that the microenvironment is the one that's most significant to the bacterium. It doesn't really care what's going on in the room, it only cares what's going on right around its cell. So the microenvironment, the most significant. Understand microbial competition, okay? Those microbes that have a slightly faster reproductive rate, they can multiply quicker, say 19 minutes versus 20 minutes, they're going to outcompete other microbes in that community simply because they're going to grow faster. So they're going to outcompete by sheer number. However, some microbes also produce a toxin or an antimicrobial product that will prevent similar-like microbes from growing in that area. We talked about this notion of succession and how it can be a very ordered and predictable succession. We mentioned milk as an example, gave you 4 microbes and the environmental changes that take place. So knowing that would be useful. I don't know, do I need to go into it real quick? Or? Okay, good. Aquatic habitats. Be sure that you're familiar or understand the concept of aquatic habitats and eutrophic conditions developing, and they're leading to a hypoxia or hypoxic condition here. So remember that aquatic systems have an upper zone and a lower zone, and the upper zone is where we're going to find the phototrophs, the microbes that are photosynthesizing. The lower zone is where we're going to find the heterotrophs, and they're usually feeding off of the algae that die and fall down here. Now, we also identified the body of water as being oligotrophic, low in nutrients, or eutrophic, high in nutrients. In most cases, large bodies of water are going to be low in nutrients, especially in the center of the body of water. Along the shelves and stuff, you can have higher nutrients, but generally low nutrients in the center of the body of water. This kind of keeps the growth in check of the heterotrophs and the phototrophs, but if you dump in nutrients, nitrate, phosphate, because that's all that the phototrophs need. They just need nitrates and phosphates because they're already getting the sunlight they need. You dump in nitrates and phosphates, these guys go into a bloom, then they die and they fall to the bottom where the heterotrophs feed on them, use all the oxygen, and create a hypoxic condition. This hypoxic condition then is one where there's very little or no oxygen, and the organisms that were there are either dying or dead or leaving that area. Okay? This can happen with industrial runoff, companies that maybe have a discharge of nitrates and phosphates and things. You can also have this happen with raw sewage, but it's a little bit different. So when you dump raw sewage in here, in addition to the nitrates and phosphates that might spur algal growth, you're actually dumping in a whole bunch of organic matter. Whole bunch of BOD. So when you put in raw sewage, this organic matter, you're really bypassing the algal bloom. You don't even need the algal bloom now because you've already put in the organic matter. So the heterotrophs feed on the organic matter that you've dumped in, you develop a hypoxic condition. So you can get hypoxia from industrial runoff, rain runoff, or raw sewage into a body of water. Any questions about that? Terrestrial habitats, a couple of the adaptations that we saw, endospores of course, some microbes producing toxins. Mutualistic relationships I mentioned, the mycorrhizae. This is a fungi with a plant, or fungi with plants. Nitrogen fixers with plants, so we talked about rhizobia with the roots of plants. And then microorganisms with herbivores, we mentioned those as well. A lot of ruminants have microorganisms in their guts to help them break down the grass that they're eating. Environmental microbiology. This was really a discussion of wastewater treatment, water treatment. You should know the three phases. You have primary treatment, you have secondary treatment, you have tertiary treatment. You should also know what's going on in each phase. The primary phase is a gravity or physical process, thank you. Gravity-fed process, physical process, removing some BOD. That the secondary is a biological process, and that this biological process really is a breakdown of the majority of the organic matter that's coming through this waste stream. And then we also talked about the tertiary treatment, the reclaim plant that we have here. Really, all I would want you to know with regards to that is that we use sand filters to get rid of the particulate matter, and then we use chlorine to kill the microbes. So in the two previous processes, we're using microbes to help us break down the organic matter, but then in the last process we kill those microbes. Understand the term bioremediation, biostimulation, bioaugmentation. Understand BOD, biological oxygen